Hello and welcome to the second recording for Introduction to Psychology. In this module, in this chapter, we will be talking about the scientific method, different types of research, and finish up with a little bit of research and ethics. I'm not going to spend too much time on this set of slides. Most of you should have already seen this in various different classes before, but again, because this is an introductory class, I do have to cover it. So first we have to look at why do we do certain things in the domain of psychology? Why do we use the scientific method, which we'll get to? And the scientific method is basically a way of assuring that what we are saying about psychology, what we're saying about a specific domain of psychology, what we're saying when we talk about specific research findings, things like that, we need to be sure that they are findings or statements that we can't be sure that they're true, but we have reasonable evidence of them being true. On the other hand, we could do just what's called armchair psychology, and that is where we just, through informal observation, speculation, we answer questions about human behavior. So in that case, what is basically going on is we see something occurring and we say it's occurring because of this without doing any research to back it up. A lot of the early methods of psychology were done through this method of armchair psychology. So we're going to talk in the next chapter about things like psychoanalytic and looking at how psychoanalytic, a lot of it was this type of armchair psychology. It's kind of where it got its name, of looking at specific behaviors being done in certain ways because that was just the way it was believed to be done without any research to back it up. However, we use the scientific method though to basically answer these questions in a scientific way rather than just a, oh, this is the way we think it is. So for the scientific method, essentially it's a multi-step technique. It's a technique where you start with doing a review of the literature. So the literature, research studies that have been published, you do a review of what's been published already. Maybe the question that you have has already been answered. Maybe the question you have has been answered but only indirectly, not directly. Or maybe it's been answered about something else but not about your specific population of interest. From there, you formulate a hypothesis. So a hypothesis, this hypothesis isn't going to be just off the wall. It's going to be based on your past experience, but also on past research that's out there. From that, you design a study. The study should be done in such a way that you reduce the possible confounds. We'll get to that when we talk about different types of experiments and how reducing other things that could be possibly explaining the behavior other than what you're studying. You then, once you've designed the study, you collect the data on that study. So you collect the data by having participants do various different things relating to what your study is. After you've collected the data, you analyze the data. So that's a little bit more on the picture that's on the right has analysis there, whereas on the left it doesn't have analysis. But after you've collected the data, you have to run statistical analysis on that. That's why statistics is so important. A lot of people dread statistics. I'm a big fan of statistics. I also teach statistics, and the analysis portion is very important because doing the proper statistical analysis will help you get to the proper conclusions. Then once you've done that statistical analysis, then you draw a conclusion. Your conclusions are based on what basically the results of that statistical analysis say. And finally, you report it to the greater field. You report it in journals. You report it in various different ways that get peer review, meaning that others are reviewing to make sure you did things correct. So there are some advantages to the scientific method. Advantage of it's very specific and it's very precise. It does it in such a way that you get to a specific goal. You do it in such a way that you reduce error. So you reduce possible error. You make the findings available for replication. That's where the publication comes in, the announcing your findings. You make it available for critique by others. And the biggest thing is those 4 goals that we had in chapter 1, describe, explain, predict, and control, the scientific method can be used for each of those. Okay, let's get into descriptive research. So descriptive research, we're going to look at 3 different main methods here. There are more methods than this, but we're going to look at 3 different main methods. Descriptive research here are going to be separate from our experimental design that we'll talk about in just a little bit after this, but these are more of methods of describing. So you can use statistics with this, you can do studies with this. These methods are just not necessarily by themselves as strong as the experimental method that we'll talk about in just a few minutes. So these 3 types that we're going to look at, first we're going to look at surveys, then we're going to look at naturalistic observation, and finally case studies. Let's start with surveys. So surveys are just a method used to obtain information by asking individuals, participants, subjects to answer a set of questions about a specific thing or about multiple things. So in this case, what's going on is participants are going to fill out questionnaires, or they're going to be asked by a experimenter a question, and then they answer it, and then the experimenter writes it down or records it with a video recording or an audio recording device. There's various different ways to do surveys. The most common method is to do just a questionnaire. So you'll find this in various different domains, but a questionnaire is related to the topic of interest that just asks questions about the topic. You might have a personality questionnaire, and the personality questionnaire is going to ask you about specific your beliefs, your behaviors, different things like that, and then from that it's going to tell you your personality characteristics or your personality traits. Survey is by far the most common thing done in psychology because it's a good first step, as well as it can be used with certain methods of analysis like correlational analysis to see if things are related. So to see if your self-report of a behavior is related to your actually expressed behavior, that type of thing. So I use this in my dissertation where I told you before in the introductory slides what I did for it, but I had couples come in the lab and they had to do a interactive task with a Confederate, but they also, after they were done with that interactive task, they went and filled out surveys for about 45 minutes. And then I used those surveys to see if there was relationships to their behavior. One of the surveys actually asked about mate retention behaviors, mate retention tactics that they do, and I wanted to see if those self-reported, so what the tactics they say they're doing, related to their actual behaviors. There are some advantages to surveys. It's quick, it's efficient, it's a very short way to get information on behaviors, beliefs, experiences, and attitudes from a large group of people. It's relatively inexpensive because if you can do, especially if you can do it online, you can get to the answers for a lot of different people in a short amount of time. You can also, because you can get such large groups, you can do a lot of demographic analyses. You can look at different things from different ethnic groups, age groups, socioeconomic, meaning how wealthy they are in comparison to those around them, cultural groups, and again, because you can get so much information from so many different people, you can use these variables as comparing variables, as variables that you can do comparisons using. There are disadvantages though. The information can contain errors. The results can be biased. How questions are worded and who asks them is important. What's going on here is that you've got various different things where people can respond in a way based on the wording of the question that might be different than what the experimenter expected. You get problems here where the wording of the questions can be culturally based. So somebody from one culture might answer or think the question's asking one thing. Somebody from another culture might think it's asking another. How the questions are worded is very important in that domain, but even beyond that, it's important in the context of just expectation and the bias that comes with that. People respond in certain ways depending on how the question leads you into it. So if I ask you a question, let's say I show you a video of an accident, and then I say, did you see any road signs in the accident? Or if I ask, did you see a stop sign in the accident? If I ask it the second way, more people will actually say yes, they saw a stop sign, than if I asked it in the first way, because I've led people into an expectation. So there are some really big disadvantages to surveys, that these biases that come in, and it's just one of the things that researchers need to be aware of. And going into it, especially when you give a survey to people from multiple different cultural backgrounds, things like that, you have to be aware that there might be errors just on the way things are worded. The next thing we'll talk about real quick is naturalistic observation. I'm a huge fan of naturalistic observation. It's one of the big things that I think there needs to be more of. So naturalistic observation is a research method in which the researchers gather information by observing individuals' behaviors in a either normal or relatively normal environment without attempting to change or control the situation. So I did my research, my dissertation research, in kind of a hybrid type thing here, where participants in this case were in a lab, but I put them in what they thought was a waiting room. So it was more of a natural setting. It wasn't a setting in which they were basically locked into a specific lab where there was specific things that they had to do. No, they just thought they were in a waiting room, so then it becomes a more naturalistic setting. So it allowed me to have more control, but I didn't have full control of it. And because it's a lab setting, though, I was allowed to bring in confederates, I could do timing, things like that. There is advantages to it. It gives you the opportunity to study the behavior in real life situations. It's been found that, sorry, I got a cat trying to rub on the keyboard. It has been found that real people who are in real life situations act different than people who are in labs. People who know they're being recorded, know they're being observed, actually change behavior. So this allows for a naturalistic, more typical behavior. However, there are disadvantages. Because it's all about observation, you could get bias in the interpretation of what's going on. What typically is done here to control for that is you have people that don't know what you're actually researching who are recording the behavior. So you don't just have it's not just a researcher sitting in an environment just watching people. It's usually people that are recorded, and then later that people code certain behaviors. One other problem is it's very time consuming, and there's a lot of training that goes into it. So my coders for my dissertation, I actually had to retrain them 3 times before they could code the behaviors in a way that was consistent with the other coders. The last disadvantage, the lack of control, means it's impossible to assign cause. Again, in a true naturalistic setting, this is a problem, but in hybrid settings or in settings where you're including like a confederate, then you can incorporate control. So that's not entirely true. Last one we'll look at is a case study. So case studies are where you've got in-depth analysis of a single individual or a few individuals. That's one thing your book gets wrong. Your book talks about it being just a single individual. Case studies are done on few individuals or a single individual. So you get in-depth analysis, you get a whole bunch of information on one individual. You record their behavior, you follow them for lengths of time. If you're doing a therapy or something like that, you give them that therapy, then you take it away, then you give them another therapy and take it away. The point is a case study gives you very, very in-depth analysis and information on a single or a few individuals. The advantage then becomes because of the detailed information, it allows for greater understanding of the participant's life. The disadvantages, though, is that because you're looking at only one to a few people, it becomes very difficult to generalize to the population as a whole, and because one researcher is doing all the collection of information on this individual, you could again get bias and stuff like that, unless of course you've got coders coding the behavior after. So all of these methods that we just talked about, they've got their advantages, they've got their disadvantages. One thing that needs to be noted, and I've kind of pointed it out a few times here, is that these disadvantages are actually able to be controlled, at least to an extent. So in those domains, the disadvantages aren't as much of a disadvantage. It's more of this is a known difficulty with this type of research design, and researchers need to be aware of it and try to overcome it. So that's where I was talking about that naturalistic observation. Most of the disadvantages can be overcome. However, to overcome them, the researcher needs to be aware of those disadvantages ahead of time. Next thing we'll look at real quick is placebos. So placebos, this is anytime there's an intervention, a treatment, a therapy, anything like that, that has no known or is known for a fact to not have an effect on, a direct effect on what it is that's being measured. So if you take a sugar pill, something like that, a sugar pill is not going to reduce pain. However, the placebo effect comes in when you actually get a change in the patient's illness or behavior that is actually attributed now to this imagined treatment rather than an actual medical treatment. So if you give somebody a sugar pill and you tell them that that sugar pill will reduce their pain, they actually feel less pain. This placebo effect is a known effect, and it's actually even interesting that you can go beyond this, because the placebo effect takes it one step farther, even when you know that it's the placebo effect is there, even when you know what you're taking is a sugar pill. If you're told this sugar pill is a placebo, but taking this pill will reduce your pain, and you're even told it's a placebo, meaning it's not actually going to reduce your pain, but you're told this pill will reduce your pain, but it's a placebo, taking that will actually still reduce pain. So being aware that you're taking a placebo still has the placebo effect. It's a very interesting thing, but when we're talking about the domain of psychology as well as biology, so if we're talking about medical settings, we have to be aware that if we're testing out, if we're doing research to see if something has a effect that we're interested in, well, one thing that you need to be aware of as a researcher is that the effect that you're finding may just be a placebo effect and not an actual effect. That's why many research designs, especially in medical fields, involve comparing to a placebo to see if the drug you're researching, let's say to reduce pain, actually has an effect greater than the placebo effect. Next thing we're going to look at now is some other domains of research where we take it a step farther and look at whether we can now start to attribute cause and effect and different things like that within our experimental design. So I talked about surveys, naturalistic observation, case studies. Well, within that you can then add in lab experiments, which we'll talk about in the next slide. But before we look at lab experiments, we have to look at correlational experiments. So correlational research, correlational experiments are when something is looked at where you're looking at a relationship between two or more events. However, if you don't do true experimental research, which again we'll talk about on the next slide, what your research ends up being is just correlational in nature. I say just correlational. I should point out that most research out there is correlational in nature. That doesn't necessarily mean it's a bad thing, it just has its limitations. So a correlational research is when we're looking at a relationship between two or more events, and you're going to get a correlation coefficient that's between -1 and +1. A score of 0 means no relationship. A score that's positive means a positive relationship. A score that's negative means a negative relationship. What does that mean? Well, in a positive relationship, as one variable increases, the other variable also increases. So let's look at weight and height. They have a positive correlation. As your weight goes up, or sorry, either way, we'll look at weight or height. As your height goes up, your weight tends to go up. As your weight tends to go up on average, your height tends to go up on average. We're not talking causal there, because increasing your weight is not going to cause your height to go up. But when we look at in individuals, as weight increases, height tends to increase, not in an individual person, but in multiple individuals. So if we look at everyone in the US, people who are heavier tend to be taller, and that's because they're positively correlated. On the other hand, we've got negative correlations. So negative is as one variable goes up, the other variable tends to go down. One instance here, even though it's not the best example, but let's look at anxiety and test performance. And actually anxiety and test performance we'll get to later is a non-linear relationship. But anxiety and test performance are such that as anxiety tends to go up, test performance tends to go down. Sorry, my screen turned off. I had to turn it back on. So as anxiety goes up, test performance goes down. So in that case, it is a negative relationship. There are some advantages to correlational research. You can provide some clues to actual causal relationships. It can help predict behavior. It's easier to do. You don't need controls. You can do it with survey results, things like that. However, the disadvantage is this assumption of cause and effect. The big thing over here, correlation does not imply causation, meaning that if you have a correlation, it does not mean that there's actually a correlation present. That's one of the big mistakes that's made in correlational research, is somebody implying that something causes something else. I gave the example of height and weight earlier. Increased height does not necessarily cause increased weight. Increased weight does not necessarily cause increased height. It definitely, gaining weight doesn't cause you to gain height. There is likely a causal mechanism there in height causing an increase in weight, but there's definitely not a direct cause between. You cannot say there's a direct cause from one or the other unless you do true experimental research. Let's get into that then. In experimental research, an experiment is when you are trying to identify this cause and effect relationship that we were talking about there. So when we do true experimental research, we're following a specific set of rules that minimize the possibility of error, bias, and chance. So that's the big thing when it comes to experiments. What we're doing when we do an experimental design is we are trying to eliminate all of the external things that could cause this error, bias, and chance. And by doing that, we can then link cause and effect between the two. We've got 7 rules that we're going to do, and we're going to look at slides for each of these. Okay, let's start with the first rule of experimental research, and that is ask. Ask is when you basically form your hypothesis. In this, you are going back to the research methods that we talked about before, the actual steps, one of those steps was hypothesis. Well, that is in one of these steps too. So first, you are asking the question. You are forming a hypothesis. And a hypothesis, again, like I said before, is an educated guess. But by educated guess, what that really important word there is educated, meaning that you've done the research on what's already out there, and you're not just basically shooting from the hip. You're actually looking at what's there and making your hypothesis based on what already exists. Then you need to identify, and this is where we're going to look at variables. So we're going to look at two different variables here. First, you've got the independent variable. An independent variable is a variable that the researcher manipulates. So if we were looking at how drugs affected pain, and we developed a new drug, and we wanted to see if that drug affected pain or reduced pain. So what we do is we design this experiment, and we need to identify the independent and the dependent variable. In this case, the independent variable, and in all cases, the independent variable is what the researcher is manipulating. So we design an experiment, and we decide that we are going to do a placebo, and we're going to do aspirin, and we're going to do our new drug. So we've got three groups. The variable that the researcher is manipulating here is what group people are assigned to, or what pain medicine people are given. So in this case, the independent variable is what is the pain medicine, whether they're given the new drug, aspirin, or the placebo. So the independent variable is what the researcher controls or manipulates. You see over here, example on the right. So in this case, the researcher is trying to see if applying fertilizer affects growth. So the fertilizer is the independent variable, the application of it. If one group gets fertilizer and another group doesn't, then the independent variable is whether their fertilizer is applied or not. The dependent variable, on the other hand, is the variable that the researcher measures. That's a good way to say it, is the outcome variable. Another way to say it is outcome. And up here we've got manipulated. So the independent variable is the manipulated variable. The dependent variable is the variable that's the outcome variable, the variable that's measured. So in our example on the right, we've got the dependent variable being growth, amount of growth, or number of leaves, or number of fruits. There can be a lot of different outcome variables that we could be looking at. In our example of pain, in our drugs, whether they reduce pain, the outcome variable is levels of pain. So the independent variable, it's what the variable that the researcher manipulates. The dependent variable, it's the variable that the researcher measures as a result or as an outcome. Next variable is choose, or the next step or rule is choose. For choose, you need to choose various different things. You need to choose your population of interest. So every person exists within various different populations. We need to choose whether we want to test all students, all people of a certain age, all people of a certain gender, all people of a certain racial or ethnic background, all people from a certain country, a certain region, a certain city. All of these are different examples of the population that we need to choose when we are doing our experiment. The population we choose is basically what we can generalize our results for in our experiment. So that example on the previous slide where they're adding fertilizer and measuring growth. Well, the population wouldn't be, you wouldn't just throw a bunch of seeds in at random. You would decide, well, do you want to measure it in a specific type of tree? Well, then your population is that type of tree. Then you take a sample. A sample is going to be a subset of the population that you are selecting to study. if your sample includes your entire population, that's what's called a census. That is rare to do, it is very difficult to do, and is expensive. So let's say our population of interest was everyone in the US. We can't collect data on everyone in the US unless it's about specific questions like on the census, and even then it's questionable whether it's collecting everyone. If our population, though, let's say is just university students in Detroit, well, maybe we can get closer to collecting everyone, but even then it's still difficult to get answers and responses from every university student in Detroit. So what we do is we get a sample. We collect a sample, a subset of the population that we are going to run our study on. And then we also need to include, when we're doing our sample, random selection and random assignment, which we'll get to on the next one. But random selection, meaning everyone in the population has an equal chance of being selected for the experiment. This is a step that is typically overlooked or skipped. Because our population is typically something greater, well, most studies are done on college students, so everyone in the population doesn't have a chance of being selected. Or if you're measuring people, let's say, that are going into a store, well, everyone isn't have an equal chance of being selected because everybody doesn't come in at the time that you're running your experiment. So it's very difficult to do, but ideally everyone that's selected should be randomly selected, meaning everybody in the population has an equal chance of getting selected. That does lead us to assignment. So now we are assigning people either to that experimental group or control group. So experimental group, and there could be multiple different experimental groups. There could be 1, 2, 3, 50 experimental groups. Typically we're going to keep it down to 1 to 3 range, but in this case, in our medication case, or our pain case with the drugs, group 1 would have been our new drug. Group 2 would have been aspirin, and then we didn't have a group 3. We did have a control group, though, that was the placebo. My spelling is horrible and my writing's horrible. I am sorry, but hopefully you understand what I'm getting at. So in the control group or the placebo group, they are a group that we are going to compare it to. So as you see over here on the right, your experimental group for this was the group with the fertilizer. Your control group was where they didn't get fertilizer. And part of assignment is random assignment. So random assignment is where everybody selected, or every participant selected for the study, or even on the case of the right, every seed in the study has a random chance of being in each group. A random chance of being in one of the experimental groups, a random chance of being in the control group. This assures that we are getting, we are reducing other potential biases that are out there. Like if we assign the first 20 people to come into the experiment to the experimental group and the next 20 to the control group, well, what if it's time of day? So the first 20 came in the morning, the next 20 came in at night. What if time of day actually affects pain? So we, by doing non-random assignment, we've actually added a confounding variable, that is time of day, in that could mess with our experiment. So random assignment is important. Then we need to do the manipulation that we're interested in. So our manipulation is where we basically look at where we do the experiment, where we manipulate it, where we either add the drug or add the fertilizer in the other example. One thing that's important with manipulation is that it needs to be double-blind. That is, that neither the participants or the researchers know what group they're in. If you know that you're in the aspirin group and not the new drug group, it might affect your behavior. If you know you're in the placebo group, it might affect your behavior. So the participants can't know which group they're in, but the researchers also can't know what group they're in. If the researcher knows that you're giving the new drug to the participant, the researcher might treat that participant different than they treat the ones getting aspirin or placebo. So both the researcher and the participants need to be blind. Even in the fertilizer example, the researcher still needs to be blind. Obviously the seeds aren't going to know what group they're in, but the researcher needs to be blind to it because maybe they know that the one got fertilizer, so they are subconsciously actually watering the ones with fertilizer more than the other ones. Or the other way around, they're subconsciously watering the ones that didn't get fertilizer more. So the researcher needs to be blind to the experiment as well. Then you measure. So you've done all this, you now need to measure the behaviors. This is where you're measuring your dependent variable. Not too much to say about this one other than when our pain example and our drugs and pain example, you're measuring their levels of pain. Maybe you prick them with a pin and ask them how much pain they received. Or more common is there's these vats of water or buckets of water that are held constant at a cold temperature, and you ask the participants to put their hand in the water and keep their hand in the water as long as they can. The cold water causes them pain. By keeping their hand in the water longer, they are showing that they have less pain, that type of thing. And then finally, rule 7 is analyze, and this is where you've measured it. Now you run your statistical procedures and you run your stats on it. So you test to see if the outcome variable is varying depending on your independent variable or if it's not. So there are some advantages to experimental research. These advantages to experimental research are things like it helps us identify cause and effect relationships. That wasn't present with correlational, so with experimental research we can identify if the independent variable is causing the dependent variable. We identify if one variable is causing the other. And it helps us eliminate these extraneous variables. Those variables that I was talking about, things like time, other different things that could be actually causing the cause and effect relationship. If we are controlling for as much as we can, if not everything, we eliminate all of these other variables that could be causing an effect. Disadvantages, first off, it's not typically representative of the population, meaning here if we are running our experiment on college students, but we're trying to generalize to the population as a whole, we can't necessarily do that. Experimenter bias comes in a lot. This is a problem where the experimenter influences the outcomes. We do double blind to reduce that, but double blind isn't always possible, and even in cases where it's double blind, the experimenter can have some bias. The raters, the ones that are coding behaviors, they could have bias, things like that. Participant expectations is a pretty big one, and this is a problem in even survey, correlational research. But if a participant is expecting to reply a certain way or respond a certain way, or the participants' expectations about research, the participants' expectations about the way research methods are conducted. Deceit, we'll talk about in just a minute, is common in research. Well, if the participant expects deceit, then they're going to second guess or analyze deeply every little part of the experiment, and because of that, they might react in different ways. Participants might react in a way that is they think the experimenter wants them to react in. So there's a lot of different things that can come up here, and researchers do as much as they can to eliminate this, but it all can't be eliminated. And then the final one is it can't be used in certain situations due to ethics. You can't assign people to groups sometimes due to ethical concerns. So you're assigning people to groups. Well, what if you're assigning people, you want to see if a certain thing causes problems with pregnancy, or a certain thing causes increases in getting pregnant. Well, you can't assign people to a group where one group is more likely to get pregnant than the others. What if you want to see if things like smoking causes birth complications? Well, you can't assign certain people that are pregnant to smoke and other ones to not smoke. So there's really big ethical concerns that come in, and we'll talk about that next. So let's look at that. Things like eat this lead so I can study its effects on your brain. Their ethics don't allow for certain things. And that is what we're basically going to look at next, and that is how ethics are conducted in the domain of psychological research. So there are definitely some concerns about being a subject, whether it's an animal subject, a human participant, all of these, their concerns come up. And due to these concerns, and historically there's been cases, instances where people and where researchers did not follow ethics. There are some very heinous examples of things like the Tuskegee experiments, where experimenters working for the US government refused treatment to people with syphilis and even gave people syphilis because they wanted to test the effects of syphilis. Taking it a step farther, the reason it was even, I mean, that's ethically wrong anyway, but one of the facts in this case that comes up that made it even more wrong is that the participants, they didn't know they were participants in research, and they were all African American. So it was in the town of Tuskegee, and the African Americans in this town were, by these researchers, again, government researchers, denied treatment for syphilis. Syphilis is something we can treat, and you just need penicillin and you can get rid of it. They were denied treatment, and in some cases they were even given syphilis by the researchers because the researchers wanted to know the effects of syphilis on the brain and other behaviors. I can go over many more examples of unethical studies that have been done in the past. And because of that, we now have some very strict guidelines in how research can be conducted due to ethics. Things that are unethical are not allowed anymore. And even when things that cause minor amounts of harm, you have to justify that minor amount of harm before you can conduct an experiment. So my example earlier where we're testing a new drug for pain. I gave two examples of types of pain. You're either pricked with a needle and measured, or you have to put your hand in a cold bucket. Neither of these cause permanent harm, but they're both causing harm. They're causing transient harm, but they're causing harm. So because of that, for any experiment to get run doing this, it has to pass rigorous ethic tests. You need to prove as an experimenter that your drug is justifiable for this pain to be caused in such that you're trying to prove that it's going to reduce harm enough in the future to the population as a whole that this temporary harm to individuals is justified. I mentioned deception earlier. Deception and debriefing are two big important components of the research process. That is because sometimes deception is needed. You can't always tell the subjects what's being studied on them because that will affect their behavior. If you tell them, if you go to a participant and say, oh, we're going to study the effects of this specific drug on pain. Well, now they know that directly you're measuring pain. It might actually cause them to change the way they respond to pain and say that they're in less pain just because they now know what you're studying. Because of that, sometimes deception is needed. I had to use deception in my dissertation research because I had to deceive the people because if they had known that I was measuring their behaviors, their mate retention behaviors, that would have affected their their expression of those mate retention behaviors. So they had to be deceived into thinking they were just there in a waiting room, that the person talking to them was not a Confederate. And because of that deception, though, I needed to do a debriefing. And a debriefing is what's after the experiment, where you basically tell them about any deception that was done. You don't even need a deception to do a debriefing. You need to do a debriefing to tell them any long-term implications of the research, any possible harm that could have been done to them, all of those things. So deception is sometimes necessary, but must be justified and minimized. And a debriefing is mandatory after all experiments. Let's look at animals real quick. There's a lot of concern about animal research. So one thing that's known, an estimated 20 million animals are used in research each year. Typically you're going to have rodents, rats, and mice, but it could be even things like cats, primates, various different things like that. There is a very big concern about ethics in animal research. You get organizations like PETA that think that there should be no animal research, but just like human research, there are ethics boards and ethics committees that must approve research to be done on animals. Are animals mistreated? There's actually only a few cases of animal mistreatment that's been confirmed. The majority of the time, animals are not mistreated. I'm not saying the experiments don't cause harm. Some of the experiments do cause harm to the animals, but it's not undue or unjust harm. It's harm that is justified. So that gets us to the question, is the use of animals justified? Because of animal research, we do have a better understanding of many things in the health fields, in both psychological as well as biological, medical. A lot of the various different things we know about the way the brain works, and different psychological disorders comes from animal research. This is where you get to the question. If you're going to cause harm in studying something, is it better to cause harm to humans or to animals? It's probably better in animals. You've got to there's ethical considerations there. At the same time, to do this research, you have to justify benefits to this research. You can't just go out and say, oh, I just want to know if animals are willing to starve to death if they're put in a maze that's unsolvable. Well, that's not going to pass ethical considerations because you haven't shown or proven that the results, the findings, are worth the pain and suffering you'd put animals through in starving them to death. So what you find is that especially in modern times, a lot of research that people want to do, that researchers want to do, are not allowed because of these ethical considerations. What you do is you get this balance between conscientious, responsible use of animals and the harm that's caused. So do the benefits to society outweigh the harm? This is a question that we ask in both human and animal research, and it's something that if your benefits to society, to people, to individuals are not greater than the harm, then an ethics board is not going to approve your research. I want to talk about one last thing real quick. This is kind of not directly related to this chapter. It's more of looking ahead, because we're going to start looking at some of the different theories of psychology. And when we're looking at theories, we need to have two things that we consider. When we look at how good a theory is, we need to consider the validity of the theory. Does that theory measure what it's supposed to measure? Does it measure what it claims? So validity is when results satisfy objectives. Validity is when it measures what it's claimed to measure. So if you have a theory that says, let's say you have a theory that says you should act a certain way in a certain situation, and you go and you measure people's behavior in that situation? Does that theory measure or justify or results of it, are they consistent with what it's supposed to measure? So that's what validity is. In validity, we usually talk about tests, specific tests, but we can also, so does that test measure what it's claimed to test? But we can also talk about it in the greater context of a theory. Does that theory measure and predict what it claims to measure and predict? Then we have reliability. Reliability is all about consistency. Are the results constant? So if you've got a test, if we're talking about a test, if somebody takes the test one day, will they have the same results a day later, a week later, a month later? As well as, does one person's interpretation, one researcher's interpretation of the results, is that consistent with another researcher's interpretation of the results? So if you take a test and a researcher has to code it, is another researcher going to code it the same way? And as I said earlier in this lecture with my study on my dissertation, I had to retrain my coders multiple times because the reliability was low. One would measure the behavior of the couple, and then another would measure the behavior of the couple, and they would get different answers. So in that case, it was low reliability until there was enough training to get the reliability high enough. Reliability is done in tests, but it's also done when we're talking about theories as well. Is what the theory measuring and predicting consistent? That type of thing. So keep those in mind when we're looking at the different theories in the slides coming up the rest of this week and next week. So in review, we looked at the scientific method. We looked at types of research. First, we talked about survey, naturalistic observation, and case studies. Then we looked at correlational and experimental research. We finished with research and ethics, and kind of a preview of what's coming next. We looked at reliability and validity. Thanks. Come on back.